students, welcome back to this video. I love to get started with positive card. Listen to this. Mistakes are proof that you are trying. Yes, you try. Success is waiting for you. Yes, our today's topic are gynoecium, carpel, then structure of ovule, female gametophyte or embryo sac.
Yes, gynoecium represents the female reproductive part. Yes, there are two conditions. First one is monocarpellary. What is monocarpellary and what do you mean by multicarpellary? Each part of the gynoecium is known as pistil or it is otherwise known as carpel. Other term is carpel. Then if there are more than one pistil or carpel is present, that is known as multicarpellary. If there is only one pistil or one carpel is present, that is known as monocarpellary. Then two more terms are there. One is syncarpus and the other one is apocarpus. See, in this condition, multicarpellary condition, if the pistils are fused together, that is known as syncarpus. Yes, in multicarpellary condition, if the pistils or carpels are free, it is said to be apocarpus. You can see this is the structure of a carpel or ovule. Sorry, carpel or pistil. Then here you can see this is multicarpellary condition. Actually, that is syncarpus. This is also multicarpellary, but it is apocarpus. Apocarpus means the carpels are free. Here, syncarpus means the carpels are united. Okay. Actually, based on your NCERT, we can give the heading instead of this gynoecium, that is the pistil, megasporangium, or it is known as ovule. Megasporangium is otherwise known as ovule. Then embryo sac. What is it? Embryo sac. Actually, this embryo sac is otherwise known as female gametophyte. Female gametophyte. See, what are they? First one is pistil, megasporangium. It is otherwise known as ovule. Embryo sac. It is otherwise known as female gametophyte. So, based on this content, we have studied monocarpellary, multicarpellary, syncarpus, apocarpus, etc. Then what about the megasporangium or ovule? Where this ovule is present. Here you can see, in order to understand all this, we should study the structure of carpel. So, a carpel consists of three important parts. They are, one is ovary, then other one is style and stigma. Ovary, style, stigma. The basal swollen portion, this portion is known as ovary. And this long slender part is known as the style. And at the tip of this pistil or carpel, we can see the stigma. Actually, this is the landing part for the pollen grains. See, this is the section of flower. Here all the parts we can see. Vegetative parts means sexual parts, or reproductive parts and non-reproductive parts we can see. See, this part is known as the corolla. Each whorl of the corolla is known as petal. This is known as the calyx. Each whorl of the calyx is known as sepal. So here we have to study about the different parts of the pistil. See, stigma is the landing platform for pollen grains. We have studied. Then style, this is the elongated slender part. Then ovary is the basal bulged portion. Okay. Then what about this ovary, or where this ovule is present? Yes, ovule is present inside the ovary. Ovule is present inside the ovary. Based on your last year's syllabus, you have studied after fertilization, ovule gives rise to fruit and ovary gives rise to ovary gives rise to fruit and ovule gives rise to seed. Yes, the position of the ovule inside the ovary. Actually, where this ovule is present. Ovule is present inside the ovary. Inside the ovary, there is an ovarian cavity. That ovarian cavity is known as locule. Ovarian cavity, otherwise known as a locule, in which placenta is located. Then, arising from the placenta are the ovules. Ovules are otherwise known as megasporangia. Yes, what about the number of these ovules? In the case of wheat, paddy, mango, etc., there is only one ovule present. But in some plants, papaya, watermelon, orchids, etc., there are many ovules present. Then what about the structure of this ovule? Yes, you can see. We have studied placenta. Actually, this is the correct position of the placenta. Yes, ovules are present inside this locule. See, this ovarian cavity. Actually, this portion is known as the ovarian cavity. Ovarian cavity is otherwise known as the locule. Then here we can see the placenta. These are the ovules. Based on this picture, there are two ovules present. Okay, next is, what about the structure of megasporangium? Ovule is otherwise known as megasporangium. So when we take the section of this ovule, we will get a picture like this. Okay, we can see the different parts of this ovule one by one. Ovule is attached to the placenta by a stalk. That stalk is known as funicle. That stalk is known as funicle. What is it called? Funicle. You can see this is the funicle. Okay, the stalk. This, if we draw the diagram, we can see the funicle in such a way that, like this. Okay, so this part means stalk is known as funicle. Then, junction between the ovule and funicle. Here there is a junction between the ovule and the funicle. That part is known as, do you know what is it? Yes, this portion. This particular portion is known as the hilum. So, which portion is hilum? Hilum is the junction between this funicle and ovule. Actually, this is the ovule. Stalk of the ovule is known as funicle. Hilum is the junction between funicle and ovule. Okay. Then inside the ovule, we can see the embryo sac. Actually, this is the embryo sac or female gametophyte. Inside the ovule, the female gametophyte is produced. Yes, while comparing to microsporangium, pollen grains are produced inside the microsporangium. Pollen grains are otherwise known as microspores that we know already. So here, the in the case of male gametophyte, actually pollen grains are the male gametophyte, while the female gametophyte, embryo sac is the female gametophyte. So this embryo sac is present inside the megasporangium. The pollen grains or the male gametophyte are present inside the microsporangium. Okay, so the development of this embryo sac that we can study later. Now we should study the structure of ovule. So inside the ovule, embryo sac is developed, and how this embryo sac is developed extra we can see later. Now we come to know the structure of ovule. Okay, what about the protective layers? Yes, it consists of two protective layers. Here we can see one layer. Then one more layer is there inside. Two protective layers are there. And it is named as outer and inner. Then what is the name of that protective layer? Of course, the name of the protective layer is integument. See, so we can call it as outer integument and inner integument. Then after that, protective layers means this is outer layer, this is the inner layer. Then inside to this integuments, you can see nucellus. You can see the nucellus. Then while talking about the integuments, integuments are absent in this particular area. Which area? Here we cannot see the integument. So that area is known as micropyle. Okay. So the part of the ovule where the micropyle is present, that is known as micropylar end. You see here you can see that word micropylar end. And the opposite side of the micropylar end is known as the chalazal part, or it is known as basal part. Chalazal pole or basal pole means there are two protective layers, outer layer that is outer integument and inner layer that is inner integument. Then there is a part, the integument is absent. That part is known as the micropyle. Actually, the micropyle is the region through which the gases reaches, respiratory gas reaches to the ovule. Then, since the micropyle is present in that part, that pole is known as micropylar end or micropylar pole. That is given here, micropylar pole. Then what about the chalazal pole? The opposite side of the micropylar pole is known as the chalazal pole. Okay, children, listen carefully. Then here this ovule is present inside this ovary. This ovule is present inside the ovary. Ovary is the part of carpel. Carpel consists of three important parts. Which are they? That is ovary, style and stigma. Stigma is the landing platform for pollen grains. Style is the stalk and ovary is the basal part. Inside the ovary, ovule is present. The location of the female gametophyte is inside the ovule. The development of the female gametophyte is taking place inside the ovule. Okay, I think it is clear for everyone. Okay, then next is I have told about the nucellus already. About the embryo sac also I have told. Then next is development of embryo sac. How the embryo sac is formed. Embryo sac is formed from a megaspore. So again we have a doubt how this megaspore developed. Yes, the very next moment we are going to study about that. So before that I have to rub all this unwanted tick mark and all. Okay. So the development of the megaspore inside the ovule, that is known as megasporogenesis. This is the term. You can see this is the term megasporogenesis. Megasporogenesis means formation of megaspore from megasporangium. Do you remember what is microsporogenesis? Formation of microspores from microsporangium. Yes. What type of cell division took place during the formation of microspores? Don't you remember what type of cell division it is? Yes, the type of cell division is meiosis, and all the megaspores became haploid. Yes, here also the same type of cell division, that is meiosis, takes place to produce megaspores. Then how these megaspores are produced? Yes, inside the nucellar tissues, you can see this is the part where nucellus is present. Yes, in the region of micropellar end, some of the cells of the nucellar tissues differentiate into megaspore mother cell. See, this is the word megaspore mother cell, or it is abbreviated as MMC. So megaspores are produced from the megaspore mother cell. Then there is a question, where the megaspore mother cell found? Yes, of course, this megaspore mother cells are found in the micropellar region of the nucellar tissue, or some of the tissues which are present in the nucellus. Actually, in that particular part in the micropellar end, they differentiate into form megaspore mother cell. Okay. Then megaspore mother cell undergoes meiosis. And how many megaspores are produced? Yes, there are 4 megaspores produced. So micropellar end, you can see here, the nucellus tissues of the micropellar end undergoes or differentiate to form megaspore mother cell. Okay, they differentiated into form megaspore mother cell. Then megaspore mother cells undergoes meiosis to form megaspore tetrad. Okay, is it clear to you? Okay, children. Then what about these megaspores tetrad? All the megaspores will be functional or not? That we are going to study. Okay, here out of these 4 megaspores, only one megaspore is functional. Means this will be degenerated, it will degenerate, then only one functional megaspore remains. From this, see this megaspore, functional megaspore. Then all these 3 are degenerating megaspores. 1, 2, 3. Only one is functional. From this one functional megaspore, embryo sac is developed. So the functional megaspore develops into the female gametophyte. Female gametophyte is the embryo sac. So it is known as monosporic development of embryo sac. Why it is known as monosporic development of embryo sac? Because it is developed from one functional megaspore. So the embryo sac formation from a single megaspore is called monosporic development. Okay. See, next, what all changes are taking place during the formation of embryo sac? Yes, again, you can see complicated structure. What about this? While seeing this diagram or photograph, it is somewhat complicated. But when you try to understand, there is no any difficulty. We can see very slowly, one by one. So from your examination point of view, this area you will get at least 3 or 4 marks. Sure. Yes, now we come to know the formation of female gametophyte or embryo sac. Here all means I'll show you what happens to this particular area. It undergoes the one functional megaspore undergoes continuous mitotic division, so that how many cells are produced and how many nuclei are produced, we can see. First, it is divided into first mitotic division. There is no any change in the chromosome number. First, during the first division, it is changed into 2. In the second division, 4. In the third division, 8 nucleate stage. How these 8 nuclei are arranged? These nuclei are arranged in such a way that we can see 4 nuclei in this pole and 4 nuclei are arranged in this pole. In each pole, there are 4 nuclei are arranged. In the next stage, what happens? One nuclei move to the center. Means from this pole, one nuclei move to the center. See, you can see the one of this nuclei from that pole move to the center. Then another one nuclei move from this pole to the center. Since they move from the opposite poles, they are known as polar nuclei. What are they called? They are known as polar nuclei. Then later they change into central cell. Then how many nuclei are remaining in this area, micropolar end? Yes, 3 nuclei. Here 1, 2, 3. How many nuclei are remaining in this particular pole, chelacal pole? 1, 2, 3. What are they called? The 3 nuclei which are present in the chelacal end, they are known as antipodals. What are they called? Antipodals. The 3 nuclei which are present at the micropolar end is known as egg apparatus. Yes, it is known as, what is it called? It is called as egg apparatus. I'll write here, listen carefully. Egg apparatus. Okay. Then the egg apparatus consists of synergid, 2 synergids and 1 egg. How many synergids are there? There are 2 synergids and 1 egg is present. Then uh, do you remember the ploidy of each cell? So here mitosis took place. Yes, before this mitosis, during the formation of the megaspore, the cell division was meiosis, so that the ploidy is N. So we can write the ploidy is N. Here also the ploidy N. Then in all this cell division, mitosis only taking place, so that here all the ploidy is N. So we can say that in this synergid, the ploidy is N. What about this egg? There also the ploidy is N. Then what about this synergid? There also the ploidy is N. Okay. Then in the case of the polar cell or polar nuclei, the ploidy is 2N. Can you say how does it come to N? Yes, it is because this nuclei is N. And here also this nuclei is N. So that it comes to N. Have you got that idea? Okay. Then antipodals. What about this antipodals? All these cells of all nuclei of all these antipodals. That means all the three nuclei means they are N. We can write here N. This also N. And what about this? That also N. Okay. So how these synergids are arranged? Synergids are found in this particular area, here as well as here in the middle we can see the nucleus. So I have to rub all this writing parts. Okay, listen. After the 8 nucleate stage, cell walls are laid down, leading to the organization of the typical female gametophyte. Yes, after the 8 nucleate stage is formed, the cell walls are laid down. Cell walls is formed, and this picture is the typical female gametophyte. Here 2 nuclei, here 3, and here also 3. So how many nuclei are there? 3 plus 3, 6. 6 plus 2, 8. 8 nucleate stage. How many cells are there? Only 7 cells are there. Why? 1, 2, 3, 4, 5, 6, 7. These 2 nuclei together form one cell. So there are only 7 cells. 7 celled stage and 8 nucleate stage. So a typical mature embryo sac consists of a typical mature embryo sac is 8 nucleate and 7 celled. This is very important from your examination point of view. A typical mature embryo sac is 8 nucleate and 7 celled stage. Yes, we can see some cellular thickenings in the synergids. Yes, this particular area. That is known as filiform apparatus. So what is filiform apparatus? Synergids have special cellular thickenings at the micropylar tip called the filiform apparatus. What is the function of this filiform apparatus? That helps to guide the pollen tubes into the synergids. Okay, so what is filiform apparatus? The cellular thickenings found in the synergids. What is its significance? That helps to guide the pollen tubes into the synergids. See, in the previous diagram, we have seen micropylar end here in this diagram, just opposite, that whatever the diagram is, you remember one important thing. In the micropylar end, we can see the egg apparatus. Similarly, we can see the antipodals in the chelacal end. Okay. Yes, this is the formation of female gametophyte. Overall of the formation of female gametophyte. Yes, you just go through this. Megaspore, mother cell, undergoes meiosis to form tetrad. Out of this four, only one is functional. Then the functional megaspore gives rise to the embryo sac. That is why it is known as monosporic embryo sac development. Then first mitotic division, 2 nuclei. Second mitotic division, 4 nuclei. Third mitotic division, 8 nuclei. From each pole, one from each pole come to the center, that is known as the polar nuclei. Okay, so here you can see a correct diagram about the carpel or pistil. So inside the carpel, actually there are three important parts of the carpel we have studied. What are they? Ovary. This portion is the ovary. If you draw the diagram, you will get a diagram like this. Ovary, style. This portion is known as the style. Then at the tip, we can see the stigma. So this is the actual diagram. Inside the ovary, we can see the ovule. This is the ovule. This portion is the ovule. Okay. Inside the ovule, embryo sac is formed. So how this embryo sac is formed? So this is the way the embryo sac is formed that we have studied. When we summarize the topic, what all things we studied? Yes, we have studied gynoecium. Actually, that is the female reproductive part. Then each part of the gynoecium is carpel. It is otherwise known as pistil. Then we studied the structure of carpel. Then next structure of ovule we studied. Then last the development of the female gametophyte. Okay, students, that's all about today's class. Thank you very much